I move away now from drug discovery more to areas which are comfortable to myself as a clinician of drug development and clinical trials, and this has its root in the Kenya program based in the University of Oxford. And you see here one of their brochures and also the various things that they're proud of having a hand in over the years, one of which is the development of Lapdap. And that's what I want to talk about next. Chlorproguanil and dapsone are the two components of this drug, and the underpinning idea was from Bill Watkins in the 1980s. We heard Bill Watkins mentioned in the preliminary introduction. And here the basic thesis was that pyrimethamine and sulfadoxine, both very slowly eliminated drugs, had been thought to be a good thing. Slow elimination means one dose only, and therefore this must be good. But it was Bill Watkins' good idea that actually this slow elimination might be a bad thing and accelerate the decline in sensitivity to the drugs. I've not been able to find any photos of Bill which are not startling in one way or another, but I thought Bridget would like to see him post-retirement singing his heart out in the choir that he now spends really quite a lot of time on. So 3 clinical trials and much lab work later, and this was WHO TDR funded at this point, and I'm grateful to Piero Oliaro, who you see pictured here as having gotten us underway. We were able to show that the new combination was more efficacious than SP. We were able to show that it offered promise as an inexpensive rescue therapy when SP had failed, and that although it was eliminated very quickly, the number of episodes of malaria in a year when you followed children up for a calendar year was no higher than it was with SP. We were also able to show, terribly important for Bill, that his original thesis was correct, that there was a greater rate of emergence of resistance with the slowly eliminated drug than with the rapidly eliminated drug. I must, while I'm mentioning faces, also pay tribute to John Horton, because all of this, as you will have spotted, rather preceded MMV at the point that I'm talking about here. So there was not that arrangement for public-private partnership that you've heard described. Bill and I realized that a couple of dusty academics were incapable of developing drugs alone, but we wouldn't have gotten very much further without a receptive drug company, and John Horton was the key to this. A very receptive TDR, Piero Oliaro, and also DFID, which provided funding. Throughout this, the Wellcome Trust remained as supportive as it had been throughout. Wellcome had funded the original work in the 1980s. Our phase 3 trials focused particularly upon safety, although I regret to say more on that in a minute. And we were able to register the drug with the MHRA in 2003 at a cost to the public health of around about 20 cents per adult treatment course, so that we were quite pleased with ourselves. We were one of the first projects, I think, to apply to MMV for funding, and I need not really set out to an audience such as this the logic underpinning artemisinin combination therapy, so I'll skip by that. Take it as read that we were expecting that the addition of an artemisinin drug would increase efficacy and reduce further the rate of development of resistance to the drug that we had already been working on. Low cost remained a very high priority, particularly since the artemisinin drugs are really quite expensive. And I've discovered that Win Guttridge, or Winston Guttridge, as I've learnt today, is also rather camera shy, but I was able this morning in my hotel room to find this particular photograph, not only of Win, but also of Brian Greenwood, without whom, again, this project would not have moved forward quite so well. And so I just want to illustrate a couple of studies. You can imagine that 10 years' work can't be summarised terribly easily, but I've picked out two pivotal trials, one of which was quite interesting, but then the next one of which killed the product. The phase 2 trial to begin with was to try to find out how much artemisinin you need in the combination drug. We discovered when we were getting underway with this project that you couldn't really answer that crisply, and so we felt that we needed to do the work again. It was important, we felt, not to expose patients to more of a drug than they needed, but it was also the case that we wanted to keep this expensive drug to as low a mass in the tablet as we possibly could. So we were looking at CD alone, together with 1, 2 or 4 mg per kg per day of artesunate in, at this stage, as you can see, a non-fixed ratio. And our primary endpoints were variable, but time to 90% parasite clearance was one of these. Really quite nice pictures then, which show that as you increase, the horizontal axis has no meaning, the vertical axis is time to a 90% drop in parasitemia. As you increase the mass of artesunate in the dosing, so time to 90% drops. And at the end of this, we were able then to fix the ratio of the 3 drugs within the tablet, and we were also pretty pleased with the rather neat peanut-shaped tablet, which broke easily. You can see that when you're in drug development, really you do become something of a geek, and you should get out more. I really did become quite enthusiastic